Next is that pure lines, like we all know that there are genetics or there are plant breeders who are doing a lot of hybridization experiments and all. Even when we talk about Mendel in your 10th class, we have done that he performed experiment with the pea plant. So in all these experiments, he prepared pure lines. So in plant breeding experiments, pure lines can only be created if autogamy is performed or self-pollination is performed. If you remember those 10th class diagrams, the F1 generation was prepared by crossing or selfing. It was crossing, and the next then this F1 generation underwent selfing to form the F2 generation. So pure lines can only be prepared if you want to perform such experiments, and that is only possible with the help of autogamy. Now, what are the disadvantages? Autogamy does not bring variation. So if there are no variations, there will not be any help in adaptation. As we all know, environment is changing. The plant has to adapt itself for the changed environment in the next year. So if reproduction is performed with the help of selfing, variations will not come, because variations can only come with the participation of two individuals. If autogamy occur, there is involvement, or even geitonogamy occur, there is involvement of only one parent. So if one parent is involved, adaptations will not come. The plant is reproducing, but it is not bringing variations which are required for even evolution to occur. Then next is, it decreases the immunity. Again, it's a topic which we are going to learn later. Then another topic we'll be learning later, that is called the inbreeding depression. Let me give you a slight amount of this information. What is inbreeding depression? When for many generations, continuously, one after another generation, selfing and pure and self-pollination is performed, slowly the progeny formed will be weak in their vigor. Vigor means that they will have very weak strength. So their immunity is coming down, their strength is coming down, agility is coming down, and their life survival also will be decreased. Vitality, all these things will be decreased because of inbreeding depression. This topic will come in genetics again. This is regarding the homozygosity, because we all know pure lines, according to 10th class also, pure lines are homozygous. If you remember those small t and capital T, that is called as homozygous. Now, what are the advantages of allogamy? So whenever there is involvement of two individuals, or definitely there is going to be genetic recombination. As recombination occurs, there will be even variations coming, because there are involvement of two individuals. So bringing variation will help in adaptation and survival, even speciation or evolution to occur in future. So as there is mingling of the genetic material of two different individuals, there is going to be a hybrid vigor. So the next generation individual will be called a hybrid, and it will be a stronger one. That's why this is called as the hybrid vigor. It will be more strong and better in efficiency. So it helps in adaptability. The higher the yield. Now next thing is, it has been found in the orchards and all, those who perform horticulture, who are into the horticulture work and all, they have performed experiment and found that in the case of pomaceous plants, pomaceous means those plants which prepare pome fruits like apple, pear and all, in these plants it has been found that whenever there is allogamy performed with the help of honeybees and all, instead of self-pollination, there is always an increase in the yield of the fruit. So somehow it is linked with the allogamy. Whenever two different individuals of plants in an apple orchard are involved, always there is increase in the fruit production. Now, what are the disadvantages of allogamy? Allogamy, there is always an uncertainty because chance factor. The pollen grain may land, pollen grain may not land. So there is always a chance factor involved. So there is uncertainty regarding the seed formation. This was the only good thing in the autogamy, that 100% seed formations are formed. Here it is never 100%, because it all depends upon luck or chance. Because an agent may fail to carry the pollen grain to the right place, or agent may be able to cause the landing of the pollen grain at the right place. Then the set of good characters. Whenever allogamy occur, allogamy means involvement of two individual plants. So whenever there is allogamy occurring, there is whenever there are good characters, remember good characters are passed on as such in autogamy. Here those good characters which are present in groups are going to be lost because of which process? Recombination. If you remember recombination of 11th class, recombinase enzyme is involved. So it may take the two good characters apart from each other because of crossing over, because genes are controlling these characters. Then there is lots of wastage of resources, energy, time and all, because pollen grains have to be prepared in large number. Then the plant has to produce good amount of nectar to attract the insects and all, even the scent, even the colorful things and all. So a huge resource and energy of the plant simply goes wasted if there is failure of the seed formation. So these are the advantages and disadvantages of both autogamy as well as the allogamy. Now the next topic to start is the types of allogamy. So for pollination to occur between two different individual plants, there are various types of agents. So this type you are going to learn is according to what is the agent of the pollination. So according to the agent of the pollination, a pollinating agent can be an abiotic or a non-living agent, or it can be a biotic, otherwise a living agent. If the agent of pollination is abiotic agent, generally there are only two types of abiotic agent. One is the wind or the air, and the second one is the water. So wind pollination is also called as anemophily. Water pollination is called as the hydrophily. Hydrophily again of two types. One is on the surface of water or under the surface of water or inside the water. This is the real hydrophily. This is not the real hydrophily reason we are going to learn in the next class. The biotic agents can be variable. If they are insects, it is called as entomophily. If there are birds, there it is called as ornithophily. If there are bats performing at the night time pollination, it is called as chiropterophily. If there are ants involved, myrmecophily. If there are snails involved in the process of pollination, it is called as malacophily. And then definitely there are humans involved in artificial hybridization. Apart from all this, even lizards and all have been found to perform pollination among many plants. So students, today you have learned about the process of pollination, the various types of pollination, what are the advantages and disadvantages of various types of pollination, and in the tomorrow's class we are going to learn in detail about various types of agents involved in pollination. Thank you.